Good afternoon and warm welcome to all the participants and to our esteemed guest. Uh, it's our honor and pleasure to have with us Dr. Deepak Upadhyay, Sir is Scientist Senior Scale, IGFRI, Jhansi, UP. And the topic chosen by our esteemed guest for deliberation today is sensors to secure health and general welfare of livestock and humans. Sir, you're audible as well as visible, and your presentation is also visible. Recording has started. Around 44 people have already joined. Please start, sir.
Okay, thank you. Thank you so much to the host of this meeting, and thank you for inviting me to present on this very pertinent topic, which is related with the livestock and human health in general. So today I'm going to deliberate upon the sensors to secure health and general welfare of livestock and human, as you can see today's topic. So first of all, I would like to first give some introduction regarding this topic, why this topic have been chosen for today's this lecture series. So as we know that in livestock, this is not yes. So we know that livestock does have a very vital role in the life of human. They provide the important livestock origin products like milk, meat, egg, skin, manure for agriculture, and whatnot. So we cannot imagine our life without the livestock. Livestock today we are means intrinsically related with the livestock. We are only depend not only dependent on the livestock are also dependent on us, and our health is also related. That is why nowadays the one health concept is coming, where it is said that healthy livestock is equal to healthy human. When we will maintain the health of the livestock, then only we can maintain the health of the human. Coming to the scenario of the livestock production in our country, everybody knows that we are a leader in the livestock population. We are also leader in the major livestock products like milk, and we are producing about 200 million ton of milk. Presently, we are owning more than 53 crore of livestock head in our country. So we in India are means accommodating most of the livestock in the world. So it is our responsibility to maintain the health of the livestock so that the population, the human population, can become healthy. We know that infectious diseases, there are so many infectious diseases which occur in livestock. We know that, like FMD, this brucellosis, HS, and whatnot. So they are the leading cause of mortality in man, and also in human. Also, there are so many infectious diseases which can cause the mortality in the human as well. So therefore, to maintain the health, we need to detect the disease timely so that the preventive measures could be taken. This is very pertinent to today's scenario that we are seeing that there is a major outbreak and the world is suffering from the COVID-19 pandemic. So in this pandemic also we have seen that the timely detection and preventive measures were playing a very important role in curtailing the disease outbreak or disease spread. So this is true for all other infectious diseases also, whether it is bacterial, whether it is viral. If we can detect the disease timely, so that we can take the preventive measure as a individual, as a nation, or as a world. There are so many tools presently in the world for the rapid diagnosis of disease in the livestock as well as human. And this rapid diagnosis of disease is very crucial. But there are some lacuna that molecular techniques like the PCR technique, RT-PCR, and genome sequencing, they need too much expertise, expenditure, and sophisticated lab facilities to detect the disease. And the conventional method like cultural method, identification method, microscopic methods, they are time consuming methods for diagnosis of the disease. So nowadays, the alternative has come, which is known as biosensors. Biosensors for the detection of the livestock disease or human disease, or other applications are also there for the biosensors apart from the disease diagnosis. But it has a say on the diagnosis of the disease also. So that is why they are also called as lab on chip, pan-site test, or point of care test. What is the importance of these biosensors? In general, we can say they are very easy to handle compared to conventional and other molecular technique presently we are using. They are user friendly and they require minimum processing. That is why they are quick and rapid. So we can timely diagnose any kind of disease with the help of the biosensors. So what is this biosensor is? Let us talk about this. So we are talking about a sensor, use of sensor in the livestock and human health. In that we are discussing that how the biosensors, we have discussed that how the biosensors can be effective alternative compared to conventional and molecular. And now we are discussing that what is biosensor. So biosensors, as we know, they are a type of sensor, but they are different in the sense that they use biomarkers. We will discuss in coming slides what are these biomarkers are to check, for example, disease, or also they can be used for the detection of stress. We will discuss. A biosensor combines a biological component with a physiochemical detector. So biosensor means it is a sensor combined with the biological component. That is why it is called as biosensor. So it is defined as an analytical tool that measures specific molecules. We call them biomarkers in any kind of sample. Say maybe it is blood, saliva, urine, etc. So they can be or worn on the body, inside the body, or they may be outside the body to detect this, to give the result on the basis of the sample collected from the livestock or human. So biosensors, they are a principle and types. We will discuss how they work and what are they made up of. So there are two things in these biosensors: biorecognition element, or say BRE, plus transducer. So in any sensor, there will be one receptor and one transducer which will enhance the signal. So in this, the receptor is the biorecognition element. So BRE may be enzyme. Mostly they are enzyme, nucleic acid, and antibody. Other from that, aptamers means oligopeptides. Bacteria, whole bacterial cells are also used, bacteriophage. But mostly in the biorecognition element for the biosensor, the enzymes, the nucleic acid, and the antibodies are mostly used. The transducer, mostly electrochemical transducers are used in the biosensors, but they may also be optical or mass-based biosensor. So EC biosensor, yani electrochemical biosensor are most common type of biosensor. And from where it has started, the first biosensor was found by Clark and Loyan in 1962 for glucose detection. So now you can understand better that what we are using the glucometer in our home for detecting the glucose level in the blood. That is a one type of biosensors which was discovered earlier in 1962, and it was the first of the biosensor which was discovered in the world. So you can imagine that how useful is this biosensor is that you just have a single prick on your finger and place the strip of this glucometer in that blood, and it will say it will within a minute it will show the glucose level of your blood. So it is that quick, and that is why we are talking about biosensors used in the livestock health also. So in this figure Let us see what we have discussed earlier. What are this theory and principles? Let us see the in this diagram also. So there are four things: analyte, the biological element, the transducer, and the processing of the signal and display of the signal. So if we are having any sample, livestock sample or human sample, that is maybe urine or blood, saliva, tear, anything or any cell, or maybe bacteria, or it may also be the sample from the environment of the livestock houses or surrounding. So the biological element, as we discussed, that it may be enzyme, it may be antibody, or it may be bacteriophage or bacteria itself, or aptamers. So they recognize these the chemical present in this sample. It may be bacteria or it may be some chemical substance. So they recognize this and they produce some. There is some kind of change, that is maybe change in the pH, maybe change in the temperature, or maybe change in the mass, which will be later detected by the electrochemical electrodes mostly, which is used as a transducer. But it may be optical, colorimetric, mass, magnetic. There are several type of transducer. Let us do not go in detail on that. But what is the function of that? We have to know that they identify this signal, the change due to the reaction between the biological element and the chemical present in the target chemical or target analyte present in the sample. So they recognize this sample and they produce a logarithm signal. Then it is in the signal processing process, it is transferred to the digital signal, and this digital signal later on will be shown in the display or the mode, which can be, which we will be understanding, or which we will see that what amount of this chemical or what amount of this bacteria is present, whether it is present or not. Further in detail, if we go, that there are several kind of the receptors, or and here I want to say that these receptors in the biosensors are means fixed on some surface or substrate, that may be glass, paper, or nanomaterial, which is recently being used, the gold nanoparticles, which are being used as a substrate for the receptor of these biological markers, or it may be silicon or it may be polymer. So but recent is the nanomaterial, that is nanoparticles, gold nanoparticles, because it offers a larger surface area to that substrate and it gives the more signal to the transducer. So this is one example of the nanomaterial based electrochemical biosensor. Here we can see these are the bioreceptor. It may be any kind of enzyme or the nucleic acid or the antibody. And here we see that there are sample. It may contain some material of the target we want to analyze. It may be glucose, it may be some ammonia, it may be some gas. So this kind of sample. So here we can say this is the layer where they are fixed, and these are the nanoparticle, gold nanoparticle. So they combine with this and they produce some kind of signal. And here this electrode which identifies this signal and present it to the display unit. For that we can identify. So this is about we have discussed about the what are the biosensors, what is the principle of their mechanism, what types of transducer are being used to make the biosensors, and what are the advances in the biosensors. So let us, in the previous slide, in the first introduction, we have discussed that the biosensors are very effective tool for identifying the animal or livestock disease, or these, or in general we can say disease. It may be of human or livestock. So for to identifying the disease very quickly, and I have quoted one example of the glucose level monitoring for the diabetic patient. So for diabetic patient, it is very easy to monitor the blood glucose level several times in a day with the use of the biosensors very quickly and with the less cost, and it is also a user friendly. Apart from that, what are the functions, what are the applications of biosensors, in which field they can be used? So the biosensors can be used to detect the bacterial infection, and we will discuss these applications one by one. Bacterial infection, water quality, viral infection detection, farm monitoring, food quality monitoring, drug residue monitoring, animal health monitoring, stress monitoring, and animal location tracing. So these are the several, or this is not an exhaustive list. Biosensors can be used in so many applications, and they are very widely used, and that is why they are being researched upon nowadays very thoroughly. So now we will discuss the different kind of biosensor research which is presently going on, and what kind of biosensors are being researched or being developed for the livestock. We will keep in center of our discussion the livestock health. So detection of bacterial infection. So there are several bacterial infection in the livestock. For example, here I am presenting the one immunoassay which was developed to identify the E. coli infection in the sample of the animal. So here this is also a biosensor. It is not necessarily it should be made up of the all the electrode and other. It may be a lateral flow assay also, where we can fit an electrode to detect the reaction chain at the last. So this is the stack pad immunoassay developed by the Altos and Marks et al. In 2017. What they have make a simple this pad, different kind of pad arranged in a sequence. We have 1 2 3 4 5 6 7. This is the first is the sample pad where the sample will be inserted, and 3 5 and 7 in between that, this 2 4 and 6, they are the separation pad. And this third is, here we can see there are antibodies combined with the horseradish peroxidase. And in this fifth layer, the bacteria E. coli, original bacteria E. coli, which is fixed to this layer. It cannot move. It is fixed to this layer. So when the sample containing bacteria will be inserted in this first pad, it will move in this direction. It will cross this separation pad and reach to the third layer, where it will be the antibody specific to the E. coli will combine with this E. coli, which is again ligated with the horseradish peroxidase. So it will go to the fourth layer, then to the fifth layer. Fifth layer, there will be the E. coli which is fixed. So there will be no free antibodies to bind with this fixed E. coli bacteria. So it will pass the fifth layer and it will reach to the seventh layer, where the reaction will be taken and the color will be developed for the bacteria presence. It will show that in the sample there is a E. coli bacteria. If there is no bacteria, there will be no reaction, and there will be. So with this example, we can understand how these biosensors may work. This is a very simple example which was means result for the detection of the E. coli in the sample of the animals. Likewise, there were several assays and there were several biosensors were developed to identify several important bacteria which causes the livestock diseases. So which I am not presenting here. Detection of viral infection. So likewise, the bacterial infection, also the biosensors were developed for the detection of the viral infection. We know there are several viral disease which are very means some are uh, very important with respect to the human health also. One of that is the influenza, avian and human influenza virus. For that Haidesima et al. 2013, they have developed a biosensor to differentiate between the avian and the human influenza virus with good sensitivity. I will discuss this in the next slide. Biosensor assay named Ventix. This is commercially developed now for the detection of the bovine herpes virus. So here I want to say that there are the biosensors which can detect the viral infections also, and they are available commercially in the market. Though presently in India they are not available, it is available somewhere in the world, but it is not available in India. But later, sooner it will come to our country also. That the research is being going on. So the detection of viral infection using the biosensor assay will be developing soon in the country also. There are several research going on. This is a simple example where sialic acid gold nanoparticles are being used. If there is no virus, there is purple color. If the influenza virus comes in contact, it will develop the red color, which will be later on detected with the help of the electrode and the signal transducing process. So here is that biosensor which was developed to detect the differentiate between the avian and human influenza virus. It is based on some principle. So this is the chip, very small chip, which can detect the influenza virus and also differentiate the between the human influenza and the avian influenza virus. So this is the 3-sialic glycan and this is 6-sialic glycan. So with the avian influenza virus has binding capacity with this 3-sialic acid, and this the human influenza virus bind with this glycan. So based on this principle, it can this is called as the field effect transistor biosensor. This is a kind of the biosensor to detect the discriminate between the human and influenza virus, and with a very high sensitivity. This sensor device can capture even single influenza virus particle displaying H5 molecule and 12 particle displaying H1 molecule. So along with the rapidity, it is also sensitive as well as specific. So which is the requirement of for any assay to detect the pathogen, that it should be specific and it should be sensitive. So it offers the advantage of sensitivity and specificity as well as rapidity. Likewise, some other diseases which are very important for the livestock like mastitis. So for mastitis, a screen printed carbon electrode has been devised to detect the anagase enzyme in the mastitic milk. This due to the mastitis, there are several changes in the milk where the anagase level increases, LDH level increases, the electrolyte composition changes, the pH changes. So based on that, this sensor can detect some of the enzyme and say that yes, it is a mastitic milk or not. Further, also it is important in the mastitis to detect the bacteria, which bacteria is causing exactly the disease. If we can detect the bacteria, we can also give the antibiotic pertinent to that bacteria only, and we can treat and we can prevent the antibiotic resistance. So, an electrochemical paper-based analytical device was developed which can detect the Staph aureus, which is very common bacteria for the cause of the mastitis. So there are several other assays also which can detect the mastitis. So they are available at present, which with the help of which the biosensors can be developed and they can be used, they can be commercialized. Likewise, the drug residue, like in meat, this is very important to detect meat or milk, detect the antibiotic residue in the animal origin food. So for the detection of several antibiotics, the biosensors have been already developed to detect the kanamycin, like chloramphenicol, streptomycin, and so many other antibiotics, which can be detected with the help of the biosensors in the animal foods. Sensors for behavior analysis. We know that behavior is related with the welfare of animal, and further the welfare is related with the health of the livestock. So to maintain the proper health, we have to take care of the welfare of the livestock. For taking care of the welfare, we have to identify the behavior of animal. We have to analyze the behavior so that we can say that yes, it is having a good welfare, or it will produce good, or it will remain healthy. So pressure sensor is a nose band along with data logger, which record the jaw movement via pressure have been developed already and being used. Acoustic analysis is used to estimate the biting and chewing movement for the analysis of food intake by the cattle. And accelerometer sensors have also been used to estimate the jaw movement and grazing. So these are behavior like jaw movement, the grazing behavior, the chewing movement. Based on this, we can that like if the chewing movement is less or grazing behavior is less, that it shows that the animal is not in a good health. So that we can take a preventive measure based on these recorded data based with the help of these sensors. What are the area we can monitor with these kind of biosensors in our dairy cow? We can monitor the temperature, the physiological parameters of the animal, like temperature, respiratory rate, etc. The grazing behavior, rumination behavior, the voice pattern of the animal, moving cough, the breath analysis using biosensor, we can identify the breath ketones. With the help of the pedometer or accelerometer, we can identify the activity, mobility, or lying, standing behavior of the animal. Or as we discussed, that milk quality and mastitis can also be identified or means analyzed using these advanced tool of the biosensors or sensors. Here is a very exhaustive list which show that there are several diseases: fever, lameness, estrus, mastitis, ovarian cyst, displaced abomasum, milk fever, retained placenta, diarrhea, pneumonia. They all can be identified using the behavioral changes with the help of the recording of several kind of sensors, out of which temperature, accelerometer and microphone. These sensors are very importantly can be used to detect the status of health in the livestock. Further, the physiological and biochemical parameters which can be identified using biosensors are breath analysis. Breath analysis for the biosensors for analyzing the infection by the Mycoplasma bovis by estimating the volatile organic compound in breath, so that the bios in this by the Alice et al. 2014, they have developed a biosensor which can be fitted with the mouth of the animal, which will identify the breath composition for the volatile organic compound, and based on their concentration, it can say that animal is infected with this particular kind of bacteria or not. Likewise, with the help of these kind of the biosensor, they are non-invasive type of biosensor, like breath analysis. There is no need for the extraction of blood or another sample. Simply by the analysis of breath, it can identify the bacteria. Analysis of metabolite in saliva and perspiration. This is also very important because saliva can be collected very easily from the animal, and perspiration also it is not very difficult to collect the perspiration. And with the help of these kind of biosensor, it will be very easy to automatically monitor the health status of the animal. Like cortisol immunosensor, which can detect the saliva cortisol level, have been developed for the quantitative analysis as well. It can detect the concentration of the cortisol in the saliva, and based on which we can say that animal is under stress or not. Alpha amylase biosensors also have been developed. Salivary glucose biosensors have also been. So with the help of the saliva biosensor, we can detect glucose, we can detect alpha amylase level, cortisol level, and so many other biochemicals. With the help of tears also means with the glucose level can be detected with the help of the biosensor, which can be fitted into the eye, where with the help of the tear it can detect the glucose. Further, in the aquatic animals, the analysis of stress in the fish have been identified, and detection of ovulation, which is very important for the reproductive management of the livestock. Timely ovulation. If we can know the ovulation time of the animal, we can determine the time of the artificial insemination, which will further increase the success rate of the artificial insemination. So for that, this is very important work. Pemberton et al. 2001b, which they have developed the progesterone biosensor. So based on the progesterone concentration in the animal, where the determination of ovulation can be done. Further, the intravaginal probe that can detect the conductivity and temperature inside the vagina of the livestock have also been developed to predict ovulation in the cat. So we have seen that the biosensors are being developed in several kind of field, like for breath, for saliva analyzing, for tears, for the determination of the ovulation time also. Here are some pictures where we can means imagine what kind of biosensor this they look like. Although this contact lens they are made for the human, but they can also be developed for the livestock for use in the livestock. This is a soft contact lens based wireless glucose sensor. So it can detect the with the help of the tear, it can detect the glucose level continuously. It sends the signal to its receiver from where the identification of the glucose level can be continuously monitored. Also, the biosensor can be fitted in the teeth for detection of several biocompound. This particular sensor was developed for the detection of the staphylococcus. And this is the example from the human side. This is the glucose watch. This is another kind of the biosensor which continuously monitor with the help of the salivary gland glucose. Oh, sorry, sweat gland present in the wrist. It will identify the glucose. Further, the biosensors, using different kind of biosensor and automating them, the network can be created with the help of which we can easily monitor the entire livestock farm. A wireless sensor network for the collection of data on breathing rate, heart rate, cattle movement have been developed by the Park and Heart 2015. A sound detection system has been developed to differentiate between the Newcastle disease, infectious bronchitis, and avian influenza in case of the poultry by the Bankar and Coworker 2016. So with this help of the biosensor, the farm monitoring operation can also be made easily. Now we will discuss, we have discussed already that several kind of biosensors are being developed or they are in the research phases. They are continuously developed, being developed, and so many researchers are working on this aspect because it is very important aspect and can change the entire scenario of the livestock industry in our country or in the world. But presently, most of the biosensors we have discussed, they are under the research phase. The sensor available presently in the market mostly are based on the accelerometer or temperature sensor. Like this is the smart, these are the product name. They are the brand names of the product. And this is the built-in sensor which they use. And what are the data they can generate? Here we can see. So this is the smart bow. It can detect the rumination activity. It can detect the heat. It can detect the location of the animal in the real time. Similarly, this eSense flash tag, cow manager sensor, tag sensor, they are also similarly used for the similar data generation. Here one Rumi watch nose bed sensor. This is a very interesting kind of sensor which use the accelerometer, temperature sensor, and pressure sensor. Here, this pressure sensor is being fitted in the halter form, and it is being fitted in the animal. So it will show the it will automatically send the data to its receiver for the measurement of the rumination and feeding behavior. So let us see in this video. Here we can see animals are grazing. The halter, this Rumi watch noseband halter is being fitted in this animal, this which is grazing in the field. So here we can say they are fitted with the pressure sensor and it generates data. For the first, it will generate the data for the this grazing pattern. Like it is generating data for the prehension bites. How many bites animal is taking while it is grazing. It is very important to determine the feed intake by the animal based on this prehension bites. Further, after the grazing, animal will go for the mastication. How many times the animal is being is masticating is also a sign of the its health, and amount it graze or amount it masticate. It will show the feeding intake or nutritional status of the animal. After the mastication and this grazing behavior, this sensor is also very useful for the determination of the rumination behavior. So this rumination, animal mostly ruminate while they are lying in this position, and this sensor is able to measure the rumination time, bout for the rumination, how much time the gulp is coming, how much time animal is ruminating. So with the help of this data, they, it will come to the our database, and we will store this data, and this data will be used for real-time monitoring of our farm livestock. We can say if there is a decrease in the rumination time, so animal might be suffering from some kind of disease. So we can again thoroughly examine that animal, we can isolate that animal and take care of that animal. In this way, these sensors are very helpful in maintaining the livestock health. So coming to the conclusion of this very brief presentation, that biosensors have a wide application in the field of veterinary science. We have seen that the biosensors in detection of bacteria, in detection of virus, in detection of several kind of diseases, behavior of livestock and whatnot. But presently there are very few biosensors which are have been commercially developed for livestock at present. And in that also in our country the limitation is very. There is no such as such we are using any kind of biosensors in our livestock operations. But and the wearable wireless biosensor system for health and digital detection are currently available in the market, as we have seen in the presentation. And from the future perspective, development and validation of the nano biosensors can open a new insight in animal disease diagnostics. So in conclusion, we can say that. Thank you so much. Thank you. Thank you for your kind attention.
Thank you, everyone. If you have any questions, please ask the sir. Participants, if you have any questions, please ask the sir. I think so.
Thank
There
you,
are
Dr.
no
Deepak.
questions.
Dr. Deepak, thank you very much on behalf of NSCL, IGFRI, and Bihar Veterinary, Bihar Animal Sciences University. I heartily thank you for a very nice presentation. And I request all the participants to directly interact with our esteemed speaker, or you may put your question or queries in the chat box for further clarification and discussion. Dr. Ravi has been patiently listening to this presentation. If you want to complement or supplement anything, Dr. Ravi, with our speaker. You need to unmute yourself, Dr. Ravi.
Hello, sir.
Yes, yes. Please go ahead, please.
Hello. Hello.
Yes,
Yes,
you
sir,
are audible.
please, you are audible.
Myself, Chavan from Maharashtra. One question is there for sir. Can nutrient deficiency can be detected by using this biosensor, sir? Micronutrient or any nutrient deficiency can be detected. Is it possible or not?
Dr. Deepak, there is a query that whether
Yes, sir.
nutrient
Yes, sir. Sorry, sir. I could not hear, sir. Can you repeat your question, please?
Sir, can nutrient deficiency can be detected by using these biosensors?
Exactly, sir. Exactly. We can detect the presence of the this sodium, potassium. The biosensors are available to detect that also. We can also detect the rumen environment also with the help of the biosensor. That is available in the form of bolus. So the sense I'm talking about here, two different things are there, sir. First is sensor and one is biosensor. So sensor is simply an electronic device which can detect any kind of movement or other thing. Or it will become biosensor when we are using any kind of the biological process in that sensor, which will identify it based on the biological reaction. Like some enzymatic reaction happens, it will change some pH or temperature or color. It will be identified based on the electrode, and this electrode will send a signal, and that will give the final display. So definitely the deficiency of any kind of nutrient, or in simple term we can say the concentration or the presence of certain kind of material in any kind of the body fluid can be detected based on this biosensor. Like glucose, this very good example, everybody is using that. So glucose is one kind of nutrient. So if deficiency of certain kind of nutrient we want to identify it, it can be very easily, the sensor can be developed, and they are being developed actually.
Okay, sir. Thank you. Thank you. Nice information. Thank you, sir.
Thank you for asking.
Any other questions?
Dr. Deepak, there is a question in the chat box. If you could access it, please.
Sensor while grazing can we calculate exact quantity of feed intake of the animal other than number of bites and yes, this is a very good question. We can indirectly calculate the feed intake of the animal with the help of the biosensors. Definitely we can calculate it. But there was only one question there.